Very good morning, my dear friends. Welcome to our CPGET free online coaching, plant sciences and botany. So today's our eminent resource person, Dr.K.RANI Madam Garu, Assistant Professor, Department of Botany, GDC, SIDDIPET Autonomous. So, Madam, please welcome and share your screen.
Okay, sir. Thank you. Very good afternoon, my dear students. So today we are going to discuss about the topic name is APOMIXIS. Generally for this minimum one question coming in the entrance point of view. Just what is APOMIXIS and what are the types of APOMIXIS?
Normally, if possible, so the APOMIXIS, enlarge. Madam, if possible, enlarge the PP.
Okay, sir.
Right.
Okay, madam. No, my mobile, sir.
No, madam. No problem, madam. No problem. Really, you're great concern about our class. Madam, please proceed.
Okay, sir. Okay, thank you, sir. So, APOMIXIS, so that is derived from two Greek words, Nana. Apo means away from, and mixis means act of mixing or mingling. So, apo and mixis, these two words are the Greek words. Apo is away from, mixis is the act of mixing or mingling. So, it is a type of reproduction. In general, apomixis it is the type of reproduction in which the sexual organs of the related structures take part, but seeds are formed without union of gametes. So please catch this point. Generally, it is type of reproduction, but in this, it is take part. The seeds are formed without union of the gametes. So normally the sexual reproductive cycle, it consists of the two important events. We know that. So in the two, meiosis and fertilization. All the reproduction, sexual and asexual reproductions are there. So sexual reproduction, it consists of first is the meiosis and after that fertilization. So in the sexual reproduction cycle, the sporophyte, it belongs to diploid condition. In turn, alternately, so it follows the haploid gametophytic phase. So it is a natural and normal process. But in some plants, these normal alternative sexual and asexual stages are absent. Please catch this point. Almost some special type of the plants, in sometimes these normal sexual and asexual stages are the absent in that plants. It happens due to disturbance in the process of the meiosis and fertilization. So even though in these plants, the living embryo, it develops inside the seed, so it is enclosed by the seed coat. So in this type of asexual reproduction, that is the seed germinate and it gives rise to plants. So they resemble the parent plant in all the aspects. So this type of seeds are called apomictic seeds. So this phenomenon is known as apomixis. Okay. It is a type of reproduction in which sexual organs are related, organs of related structures take part, but seeds are formed without union of the gametes. So for the first time, it discovery of this phenomenon is credited to Leeuwenhoek, Antony van Leeuwenhoek, as early as 1719 in the citrus seeds. So it belongs to Rutaceae family, all the citrus seeds. So this the genotype of the embryo and resulting plant will be the same as the seed parent. So this is in general, it is the clonal seed production. Okay. So almost this is the only it happens due to disturbance in the process of meiosis and fertilization. So in this type, asexual reproduction, the seeds germinate and give rise to plant. So it resembles the parent plant in all the aspects. But in this type, so the seeds are called apomictic seeds. This phenomenon is known as apomixis. So it was first discovered by Antony van Leeuwenhoek. So in generally, almost it is in 1719, but before that, Astonfield Rambier. So Astonfield Rambier, he is also discovered in some partial way. So that is plant Taraxacum and Hieracium plants. So Taraxacum plant first discovered by Astonfield Rambier, but he not explained proper way. After that, Antony van Leeuwenhoek. So he is in the 1719. So in the citrus, almost rutaceae plants, he discovered that the citrus roots. So the genotype of the embryo and resulting plant, it will be the same as the seed parent. Okay. So in this almost this process usually occur in the plants belonging to Asteraceae, Poaceae, Rosaceae, Rubiaceae. So these families, some like Asteraceae, Poaceae, Rosaceae, and Rubiaceae plants, these all are having phenomenon. This process. Sir, are you able to hear my voice?
Yes, madam.
Hello?
Voice is audible, madam.
Hello? Oh, okay.
Madam,
One
your voice
call
is clearly audible.
is coming.
Okay.
So,
Right.
okay, sir. Okay. So, almost the some special plants like they can produced by apomixis. It resembles the parent genotype in all the aspects so far. But apomixis is recognized in the plants belonging to 35 families of the angiosperms, Nana. According to Kush, so he is almost discovered that the apomixis, it is recognized in the plants belonging to 35 families of the angiosperms. So just like the familiar families like Asteraceae, Poaceae, Rosaceae, Rubiaceae, these plants, this process usually occur in these families. Okay. So in these apomictics, they are more common in polyploids. Almost apomictic plants more common in the polyploids than the diploid plants. So the term apomixis was coined by Winkler. Almost this is in coming. The usage of this term by plant breeders, so that is confined to only asexually formed seeds. Asexually formed seeds. So that's why this term belonging to only the apomictic plants. And in these plants, the consequently this apomixis process of that is divided into two categories. So they are vegetative propagation and agamospore. So in this way, in the apomixis plants, the development of embryo without sexual fusion. So sexual life cycle is the short circuited. Almost the development of embryo without sexual fusion. So in this, the sexual life cycle is short circuited. In this, the genotypes of the developed plants are identical to the parental plant. Okay. So almost as I told you that it is discovered by Leeuwenhoek in 1719 in the citrus seed. Okay. So the mode of reproduction, the sexual and sexual product as common plants. So sexual reproduction, the reproduction and apomixis. Apomixis is the gametophytic and sporophyte. Almost they are the vegetative. The consequently, the process of apomixis is divided into two categories. They are the vegetative propagation and agamospore. So just like vegetative reproduction and apomixis. So we know that almost vegetative reproduction And now we are entering into that apomixis. Okay. So vegetative reproduction means the propagation in this method, the apomictics, they are developed either from parental seeds or from plants. They are originated from other parts. Hence, they are called as a propagules also. Vegetative propagation, plant heads are just two parts is called propagules. Okay, they are developed from parental seeds or from the plants originated from other parts, not the sexual parts, or they are not participating in the anther and almost style and stigma, only the vegetative parts. So hence these are called as a propagules. Okay, and so this is the mode of reproduction. Like asexual, after sexual reproduction kids are formed, so that is normal reproduction. But in the asexual reproduction, it is divided into two, apomixis and vegetative. So vegetative, already vegetative propagation is completed, and next apomixis. So in the apomixis, almost divided into sporophytic and gametophytic. So the gametophytic also divided into apospory and diplospory. So in this way, almost now we are discussing about that the agamospory. So this is the almost the types of apomixis. So it is unused cells gives rise to a megagametophyte. The types of apomixis, gametophytic apomixis and sporophytic. So inside discuss that gametophytic, diplospory, apospory and embryony, parthenogenesis, and apogamy. So the slight differences, almost in the sporophytic, unreduced cells gives rise directly to an embryo in the sporophytic way, or sporophytic apomixis. Gametophyte cells raised to a megametophyte. So the embryo sac, it is originates from the megaspore mother cell. So that means in normal process of the reproduction. Gametophytic means normal way. So in last 1 week, we'll discuss that almost the microsporogenesis, megasporogenesis, and development of the embryo after fertilization, double fertilization, triple fission. These all are including in that gametophytic way. So the embryo sac originates from the megaspore mother cell either directly or mitosis, and after interrupted meiosis, the embryo sac originates from nucellular cells. The most common mechanism of apomixis in higher plants, it is character embryo sac. So in between, so in between some semigamy. So it's nothing but in between of the gametos and sporophytic. Semigamy means the haploid sperm nucleus enters the egg but does not fuse with the haploid egg nucleus. So in this, each nucleus divides independently, it creating a haploid embryo that contains the sectors of male and female origin. This is some types of the apomixis. So now the gametophytic apomixis. Gametophytic apomixis, that is divided into diplospory and apospory. So if the unfertilized cells, it gives rise to a megagametophyte. Gametophyte means unfertilized cells. So if the unfertilized cells give rise to a megagametophyte, it is divided into two types, diplospory and apospory. So in the diplospory, megaspore mother cells give rise to a megagametophyte, but in the apospory, so the embryo sac originates from the any diploid cell except megaspore mother cell, either directly by mitosis and or after interrupted meiosis. Okay, so in this generally the so and another, so that is the androgenesis. Androgenesis means the embryo sac originates from generative nucleus of pollen tube cells. The most common mechanism of apomixis in higher plants, it is characterized by the presence of multiple embryo sacs. So this is the sporophytic apomixis. In the sporophytic apomixis, it is the same. If the unfertilized cells give rise to directly to an embryo. But what is the difference between? So unfertilized give rise to a megagametophyte, that is the gametophytic apomixis. If the unfertilized cells give rise to directly to an embryo, so directly to an embryo, that is the sporophytic apomixis. Okay, so in this almost in the sporophytic apomixis, haploid parthenogenesis are there. So the embryo developed from the egg cell without fertilized egg, so that is developed as an embryo. Haploid parthenogenesis means embryo developed from the egg cell, that is the haploid parthenogenesis. And haploid apogamy, so in this it is also known as pseudogamy. The pseudogamy, embryo developed from synergids or antipodal cells. So we know that almost in the embryo, in the ovule structure, 2 synergids, 1 egg cell, 3 antipodal cells, 2 polar nuclei or diploid nucellus is present in the center part. So in that, the embryo is developed from the 2 synergids or 3 antipodal cells, that is the haploid apogamy. So that is nothing but pseudogamy. And in some other way, so adventitious embryony. So it is sporophytic budding. In this, the embryo directly developed from nucellus or integuments. In this, no production of embryo sac. For example, as I told you that some Rutaceae family members, Rutaceae and Anacardiaceae. So just like particularly mango and citrus plants, they can develop from the adventitious embryony. So in this, the embryo directly developed from nucellus or integuments. You can see here, this is gametophytic and sporophytic generations. The sporophytic generation is in the 2n stage, but in the gametophytic generation, haploid is. So stage 1 alternative generation is occur in some special plants like Rutaceae and mango plants. So the sporophytic generation, almost the megaspore mother cell, it will produce some somatic cells. So somatic cells, ovule, it will undergo the meiosis and haploid mega spores. So some megagametophyte is present. So some microgametophyte, both to fuse, undergo the fertilization, double fertilization and formation of zygote. Zygote will produce the embryo. Embryo comes seed, seed come plant. Plant will produce some megaspore, mother cells and somatic cells. So this is alternative generations. So this is the types of apomixis based on occurrence. It is the non-recurrent type and recurrent apomixis. So in the non-recurrent, embryo developed from any cell of the embryo sac, just like develop from egg cell or develop from synergids or develop from antipodal cells. These 3 ways, that is the non-recurrent apomixis. And in the recurrent apomixis, the embryo develops from diploid cells. So almost we'll discuss diplospory, apospory. Embryo sac developed from megaspore mother cell, that is the diplospory. Embryo sac developed from any vegetative cell of ovule, then embryo is developed from the diploid egg cell. So that is the apospory. Any vegetative cell, please difference between diplo and apo. Diplo develop from only megaspore mother cell, okay, without meiosis from diploid cell, especially megaspore mother cell, that is the diplospory. And without meiosis from diploid cell, develop from any vegetative cell of the ovule. So then embryo that is developed from the diploid egg cell, that is the apospory. So same adventitious embryo directly developed from nucellus or integuments. That is almost we'll discuss last slide. And what is the advantages of the apomixis in plant breeding? So what is the use? What is the significance of the apomixis? Okay. In this, just like applications and advantages of apomixis, in this, the scientists, they are considered that these apomictic as evolutionary end as they lack reduction, division, and fertilization. But today it is noticed that there are advantages or applications with apomictics. So the apomictic plants, they can resemble the parent plant in almost all aspects. Okay. So hence, by this method, many plants with the same genotype can be created in this way. Okay. So in this, almost the rapid multiplication of genetically uniform individuals, it will be achieved without risk of the segregation. Okay. Individuals can be achieved without risk of segregation. In this, some heterosis or hybrid vigor can permanently be fixed in crop plants. There's no problem for recurring seed production of F1 generation or F1 hybrids. So at the same time, the sectors retain many generations also. So even there is in hybrid vigor, by apomixis, one can produce plants in short period, which are similar in form and characters with the hybrid plants. So which are obtained through the complex hybrid experiments. Okay. So in this, almost the efficiency. 5 minutes. Okay. So the apomixis, it is not frequent in crop plants. Hence, some scientists, they are trying to put offer their efforts for the increasing crop yield by creating various kinds of the apomixis. So in this, the efficient exploitation of maternal effect, if present, that is possible from generation to generation. In this way, some homozygous inbreed lines, as in corn, it can be rapidly developed as they produce sectors of diploid tissues and occasional fertile gametes and seeds also. So some advantages of apomixis, it will be obligate apomixis. It permits fixation of heterosis in the hybrids. Therefore, farmers can resow the seeds produced by apomictic hybrids generation after the generation. So the new hybrid variety could be multiplied from new hybrid seeds in the same manner as pure lines. So this greatly simplifies hybrid seed production. And even such parents that flower at different times may be crossed in a greenhouse to obtain few hybrid seeds. So it can be used to establish the new hybrid varieties also. The nucleus seeds, seed or hybrid varieties can be conveniently maintained as hybrid varieties. So just this is the almost in all the aspects of some vegetative growth phase, floral initiation, stamen and gametophyte, ovule and gametophyte, primordia will produce. So primordia will just modify is diplospory after the meiosis is absent. So almost the meiosis, double fertilization, zygote, proendosperm, seeds, these all are absent in this way. Just primordia will come diplospory after adventitive proembryony, apospory. So these all are occur one by one in this way. But what is the methods of identification of apomictis? So the methods, just like there are many methods and techniques to identify the apomicts. Some of the in that, first is the apomictic in plants can be identified by cytological analysis of developing embryo cell. First be, so the apomictic in plants, it can be identified by cytological analysis of the developing embryo sac cell. And second, so only 3 cells, they are parent in the aposporic embryo sac. So while the normal cell number is 4, almost 3 celled egg apparatus and 1 is the center cell. So in that way, while the normal cell number is 4, this condition can be identified by the sections of tissues. And after that, so during some megasporogenesis, the callose layer is formed in apomictic embryo sacs. while in normal embryo sacs, there is no callus layer formation. So that means the formation of callus layer is, it is also identify the apomictic type. During the megasporogenesis, some callus layer is formed in the embryo sac, but that is formed only the apomictic embryo sac. But in normal embryo sac, there is no callus layer. So this way, this is also one identification method. And some variability that is seen in the heterozygous plants, so it will be obtained by cross pollination. In sometimes, though the plants are obtained by this method, if all the plants resemble the parents, they can be considered as apomictic, definitely. And some non-apomixes, so it can be easily recognized and separated, which are obtained in the breeding of progeny by restriction fragment length just to RFLP. So restriction fragment length to polymorphism. So it is a new method in the breeding of the plants. So these all are the methods of identification of the apomictics. Okay. So sir, Tirupati sir. Hello. Sir, I have just a small work is there. Principal sir call me urgently. Hello, sir. Sir, are you there? Are you hear my voice? Sir, are you able to? Sir, please.